As part of the evolution and comparative genomics track, I will present a poster entitled TAGOPSIN, a tool for retrieving taxa-specific protein structural and functional information. My name is Eshan Bundhoo and I am a graduate student at the University of Mauritius. So, with advances in experimental techniques and digital technologies, there is currently a wealth of biological data readily available in public databases. For instance, databases of the NCBI, UniProtKB, PFAM, ISCB PDB, to name a few, are specialized databases that store different types of information. The amount of biological data thus available is growing at an ever-increasing pace, so much so that this has greatly simplified the task of researchers needing select formatted data for a particular investigation. However, data retrieval is too often done repeatedly in an unsystematic way and on an individual basis. The need thus arises for a systematic way of retrieving and integrating public information available for a specific taxon. Many biological research questions can only be answered by combining information from a number of sources. We thus increasingly need to query and retrieve interconnected data from multiple databases to tackle a research problem. For example, if we want to analyze coding sequences of a given gene product for a set of related organisms, we might need to combine data stored in NCBI taxonomy with data stored in NCBI GenBank. But this process can prove to be cumbersome, and to the best of our knowledge, there currently exists no tool that does the task of assembling data from multiple databases into a single repository for ease of data manipulation. We thus designed and propose here TAGOPSIN, a program written in Java whose purpose is to retrieve select data from 7 of the most popular biological databases and integrate them into a single data warehouse managed by PostgreSQL. So these databases are NCBI Taxonomy and GenBank, UniProtKB, Gene Ontology, PFAM, EBI SIFTs, and ISCB PDB. To illustrate the features of the software, I've reproduced here figure 1 of the poster. The input to the program is an organism name specified at the command prompt. Given the organism name, TAGOPSIN retrieves taxonomy IDs and scientific names from NCBI taxonomy database. Once taxonomy IDs are known, in the subsequent steps, data retrieval from GenBank, UniProtKB, PFAM, Gene Ontology, and ISCB PDB is restricted to only those IDs of interest. And in the process, at each step, TAGOPSIN concomitantly inserts all data into a relational database in PostgreSQL, such that at the end, the output of the program is organism-centered data organized in a data warehouse in PostgreSQL. The relational database schema, figure 2 of the poster, shows the set of data retrieved by TAGOPSIN from the FTP or HTTP server of each individual database, data that we thought would be most relevant to a research study. The arrows and some boxes indicate how one database is linked to or mapped onto another database to really form one unified repository with all data in one place. Now, TAGOPSIN was tested on a Linux Ubuntu system with a number of prokaryotic organisms. The results, outlined in Table 1, indicate that the tool shows satisfactory performance and succeeds in doing what it has been designed to do, that is, retrieve data for each entity type listed here in the first column. I would like to stress the fourth column, Clostridium, which reveals that the genus name can also be used as input to the program, in which case it will retrieve data for all available species of that genus. I've used as example a subunit alpha of tryptophan synthase involved in tryptophan biosynthesis to demonstrate the added value of the tool. In the UniProt database, for instance, you don't necessarily have the coding sequence of an individual UniProt entry. So, by integrating NCBI GenBank with UniProt KB, TAGOPSIN manages to retrieve the coding sequence of each gene product in the column seq here, and these coding sequences can be very useful in a comparative genomic study, like here for different strains of Streptococcus pneumoniae. To conclude, I would say that TAGOPSIN's main originality is that it integrates organism-centered data into a data warehouse system. The data assembled can be utilized on a user-defined basis, and TAGOPSIN really constitutes a valuable tool in molecular evolutionary and other comparative genomic studies, as well as structure-based studies. That's it. If you would like additional details, please refer to the poster.